automated oligo design pipeline. And I guess unlike all the other talks so far, I'm looking at environmental sequencing, and this application came from wanting to know if my tax of interest was in an environmental sample, especially when we're looking at quarantine and regulated species. They're defined at the species or subspecies strain levels, and a lot of the similarity-based tools for metagenomics aren't really capable of this resolution, especially when we're using amplicons. So the concept for AODP is that we generate signature oligonucleotides, or SOs, based on a FASTA and a phylogenetic tree. These SOs are short nucleotide sequences. They're specific to either sequences or clades within a tree. So this means they can be specific for several sequences, a taxonomic group, and various levels of resolution. And we've also incorporated options for thermodynamic calculations. And this allows these signature oligonucleotides to be able to be used for primer design. Similarly, we've added the ability to filter out regions of the DNA that only have a single SNP. This helps us improve our ability to make primers and also to get rid of some of the noise we see in NGS datasets because of sequencing error. In our studies, the false positives usually came from a single nucleotide error during sequencing. The advantage of AODP over some of the other methods is that we can use any set of DNA sequences, and this avoids the need to use public databases, which we all know are kind of messy. So this is the overall workflow for AODP. So we start with a sequence file and a tree. The tree is optional, but it does give you a lot more information. And then we use several options from the users, such as any thermodynamic properties you want for primer design, any limitations on homopolymeric regions. Homopolymers cause problems with primer design and for next generation sequencing technologies. Whether we want to limit the number of SNPs in the DNA region, and whether we want to do any further cross-validation of the SOs against other reference databases. This will help improve our specificity depending on the use cases. AODP builds a non-blocking tri-data structure and generates the SOs and produces various outputs, FASTA, GFF, TAB, visualizations of your tree. So the AODP algorithm is based on this non-blocking tri-data structure that's built specifically for DNA sequences. So this is A, T, Gs, and Cs. This makes it much simpler and more complex at the same time because you're not considering the rest of the alphabet. And it scales well to multiprocessor architectures because it has inherent collision detection. So unlike previous versions of this software where we couldn't run on a small data set in weeks, we can now produce signature oligonucleotides for the entire UNITE database in just over an hour if we include the thermodynamic calculations. A data set, say like ITS sequences for penicillium, run in under a minute. So I guess it went ahead without me. SOs can also be produced with minimal memory footprints. Memory was also a problem in the previous version of this software. The UNITE database takes 25 gigs of RAM, while the ITS data set for penicillium takes 18 megs of RAM. So this is very scalable to the desktop. Some of the use cases for the signature oligonucleotides and AODP have been to use them directly for in silico identification of taxa from environmental NGS data based on sequence identity. We've used sequence identity to either verify the presence of quarantine pathogens in environmental data sets or to challenge an identification by a classical pipeline. Sometimes a lot of the public databases are really heavy in quarantined and regulated species, and so you get those as false positives, and using sequence identity, we've been able to say, no, this is actually a false positive, and that's good when you work for the government. We've also been able to detect candidate DNA barcode regions for taxon groups and assist with taxon-specific primer and probe generation for diagnostic assays. Most recently, we've been approached about using AODP to identify divergent regions of the genome for using comparative genomics. We've been able to design primers that are able to detect closely related genomes. So lastly, I'd like to acknowledge the bioinformatics group at AAFC. Dr. Kovis Visagi and Keith Seifert for providing data sets during our testing, and the lab and my supervisor, Dr. Wen Chen. And this is our lab. Oh. And you can come see me at poster B02. That's our software repository and contact information. Thank you.